Thank you all for coming. I hope that this talk is not too boring for the ecologists or people in evolution, but it's molecular biology. Bear with me. I tried to come up with a title that is putting together the project that I worked during my PhD, but also the project that I'm working here at NIMBioS. In both of those projects, I'm working with the same pathogen and the same molecules. However, these projects are totally different, and you will see why. I'm working with Trypanosoma cruzi, and the first project is to develop an algorithm to characterize GPI-anchored proteins. And the second is to use these GPI proteins and GPI-anchored proteins to predict epitopes. So Trypanosoma cruzi is the causative agent of Chagas disease. Chagas disease is one of the neglected tropical diseases, and it's caused by this parasite, by T. cruzi, and is transmitted by the triatomine bug, or the kissing bug is what they call. And what happens is that the kissing bug takes a blood meal from the host, and then at the same time it poops on the host. And then the host scratches, and just with a small wound, the parasite enters the body and goes to the bloodstream and initiates its cycle and remains in the body for several years undetected. I will explain why, and this is a very interesting thing. And it causes, throughout the years, cardiomyopathy and gastrointestinal disease. It is the endemic zones for Chagas disease are these 21 countries in Latin America. However, it has spread all around the world, of course, including US. So there are 15 to 18 million people infected in Latin America alone, and 300,000 is considered that are infected in the United States. From all people, 100 million are at potential risk of infection, particularly in rural areas and very poor areas. 14,000 die a year, each year. 14,000 die each year for this disease. And T. cruzi not only infects humans, it infects other species. I put here the 100 species of non-human mammals, but also infects chickens or other birds or things like that. So this is a very interesting parasite. It's a unicellular parasite, and it infects a lot of species. Treatment for early infection exists, but it's very expensive. And current drugs have very severe side effects. And there are no preventive vaccines or prophylactic drugs to prevent the infection, but I will come up to this point later. One of the main targets for developing a vaccine or a drug are what is called variant surface glycoproteins. The parasite is a unicellular parasite, and is covered with a thick layer of these variant surface glycoproteins. And this is exactly what makes the parasite so good at being undetected by the immune system, because it changes the configuration of the molecules in the surface every time that the host is already developing immunity against the parasite. This changes the configuration, and it remains undetected for years and years, sometimes even more than 10 years. So it's kind of like AIDS in that sense, that can be there for a long time, and that's when it causes these horrible side effects or these horrible diseases. At the beginning, the parasite, what it causes is just like a flu for the first 2 or 3 weeks. So people who has the parasite, who has been contaminated with that, is going to develop just some kind of flu symptoms. So then it can be detected on blood, but people is not going to be tested for that because it's going to be confused by that. So one of the main variant surface glycoproteins is this molecule, glycosyl phosphatidylinositol, or simply GPI-anchored or GPI. And this is exactly the name, an anchor that puts a protein in the surface of the membrane of the parasite. It's composed of a glycan group and a lipid group, the anchor. To construct this anchor is a post-translational modification, so it's not under direct genetic control. except the protein, of course. That one is under direct genetic control. And it requires a chain of enzymes all acting in consecutive way to form this anchor. And then attached to the anchor in the C-terminus of a protein is going to be all this other structure, all this glycolipid. I want you to keep in mind, please, the anchor part where the protein attaches to the entire molecule. The protein attaches in one amino acid, and this one amino acid is called the omega site. This omega site is important for the molecular biologists who are studying how to develop a vaccine or a treatment. And I want you to remember this for my project that I'm working here, because it's not the same to have a glycine than an isoleucine or any other amino acid. They want to know exactly which amino acid is in this omega site to do their job. These GPI anchors have a very conserved structure generally, consisting of, beside the protein, it has ethanolamine phosphate, at least 3 hexos. It has phosphatidylinositol and 1 or 2 fatty acids. The fatty acids can be of 4 different types. And these are extremely diverse because it can contain, for example, in the fatty acid, a degree of unsaturations and different number of carbons. The sugars that they have, they are a lot of different sugars, and they have a different number of those sugars. They can exist not only on Trypanosoma cruzi. Even us, we have those molecules in some places of our bodies, particularly in the brain. However, it has been shown that these molecules are overexpressed in ovarian cancer or in breast cancer. So these are important for that. And also insects also have that, such as Rhipicephalus microplus. The identification of these molecules is not easy. It requires a lot of experience by a mass spectrometry analyst. They take months and months to look for patterns and recognizing these molecules in the spectra. There are a few different strategies to do that. And basically, this is dividing the analysis just for the glycans in 3. However, you can put that also the lipid. They have a biological sample. They digest this biological sample with some enzyme. Most of the time, they use trypsin. They divide this protein into glycoprotein into just peptides. And in the case of intact glycopeptides, they perform mass spec analysis and they do data interpretation. This one is mostly done manually. The second one is to the glycosylate these proteins or peptides. They do mass spec, and then you run the peptides to a database, and then the database tells you which proteins are most likely to occur, and this is how they do protein sequencing. And the third one is taking only the glycan, do the mass spec, and do the data analysis and glycan identification. There is some software for only the glycan or for only the lipid, but as you can imagine, the software that is in use for glycolipids is very scarce compared to the ones that exist on proteins or genome. So there is no software existing right now available for identifying these GPI-anchored molecules. And this paper, this is the only paper that describes how to characterize the entire set of GPI molecules within an organism. And that's why they call GPIome, GPIome, genome, proteome. So they come up with this, of course, on T-cruzi, and because T-cruzi contains a lot of these, and they come up with 79 different GPIs. They repeat itself many times in the surface of the parasite. And just to give you an idea of why is this complicated to determine the structure of and to characterize these molecules, I want to show you. This is how they, in the mass spec, is the protein fragmentation or the peptide fragmentation. Usually the peptides, how do you break each amino acid after the next are these ions, the B and Y ions, and then you have a sequence of different amino acids. However, in glycans, for example, is much more complicated because you have the anomericity and you have branching, because glycans form branching. So it's even harder to look at the spectra and to characterize a glycan. And the lipid is even also difficult to do it. I put here in this picture a spectra in which it is characterized this GPI molecule. Here you can see these peaks corresponding to fragments, and some of those have the different sugars, hydrogen, water, and so on. And this peak, and only this peak, is the lipid. So this is the lipid here of the molecule, and characterizing this with this spectra is hard, and it requires a lot of resolution of an instrument. So what they do is that they fragment again, and that's why they do MS3. So they have to fragment one more time to come up with a very good description of the lipid. Also, to complicate even more, there are the GPIs are very isobaric. So, for example, these two structures have exactly the same mass. However, this hexose is in a different position than in this hexose, and you can see this mass here and this mass here is the same. And this mass, 1837.82, is almost the same as 1837.86. So the difference is minuscule, and you can see that the lipid is different here in reference to this one. So having isobaric GPIs makes it difficult also to characterize these molecules. What I did was to develop an algorithm based on protein characterization. And what I did was to first build a GPI library. This is a theoretical library because there is no information on this in the web or anywhere. There are just a few structures recognized. And to build this library, I used the knowledge that we have on the molecules, like the general structure that we have and how many hexos can we have, how many different sugars, lipids. And I come up with 1.9 million different structures for doing this. Then I filter every spectra that is from the experiments, looking for the hexos. And if it contains hexos, then it's a good candidate to be searched against the library. And in that case, I eliminate a lot of spectra that is not going to be good. Then I start to match fragments with first of all the parent ion mass. That means the entire mass of the molecule. I match to see how many structures are in this library, and then I match the fragments and see, and I give them a score to each one of those. And at the end, I have a ranked list of structures and a probability of occurrence. Because it requires a lot of searching and looking and building theoretical spectra, I have to do it I did it in two ways. First, I did this as a serial algorithm, and then I did it as in parallel algorithm. For that, I used three systems. I use three systems for several reasons. One is this HT Condor. This one is very easy to program because this language itself can use the resources very intelligently and use the idle cycles of computers that are placed in the pool. So that means that if some user is doing something in the computer, then the job that you submit to the pool is going to wait until the processor is idle again and then is continue working. So that makes it a little more slow, but it has the advantage that you don't have to program a lot. In the other two systems, I use MPI. This Texas Advanced Computing Center, I started doing this, and then when I was about to conclude my programming, they placed this on my institution. So they asked me, okay, instead of here, put it here, so I have it in 3 systems. And both of them use MPI. At the end, what I had, my testing data was containing this number of spectra. I filtered this number, and I was able to identify 78 structures out of 79. One of those is very difficult to identify because besides all the glycan, lipid, and everything, it contains also an adduct. It contains sodium. So placing the sodium gives a little more noise, and it's not easy to know exactly where the sodium is going to be placed within the molecule. And to compare the algorithms, I try to use some Proteome software, something that is already there. Because there is no software for GPI characterization, I try to use something that is for proteins. So in the serial part, I use 4 cores on a Linux machine with 24 gigs of RAM, and the data library of 1.9 million structures, and it took 10 days to process. 10 days is a lot of time, but it took months for the specialist to characterize this same set of structures. But if you see the proteomic software, something that is very standard is the Proteome Discoverer. This is from the company called Thermo. And we have this in a machine with 2 cores, 48 gigs of RAM, and running on Windows 7. And for characterizing the GPI proteins, there were this set of proteins with different parasites. And it took 3 days and 19 hours. So if you compare the number here, I think this does a bit better. But to do it even better, I use the efficiency of parallelization. And this is the serial one, 10 days with 4 cores. With HT Condor, I started with 72, and I did more or less these days. However, I was able to reduce even more to 24 cores, and then it takes 3 days and 10 hours with this system. With HPC, it takes 1 day and 5 hours, and with this, it takes 21 hours. So I think it's very good. And if we put more more machines, sorry, in the pool, then I think it's going to be even better. I still work on this, so my data library is a work in progress. As more information is available on more sugars or something in the molecule, we can have better versions, but of course it will have more molecules in that. And I developed just a few months ago the new version, and it contains 5.4 million structures. Then I collect. One other project is to collect and classify these GPI molecules in a database, so people can access and see where they come from. Right now, everything is scattered in papers and that because there are not so many. But one of the projects is this. The other is, of course, to extend the functionality to be able to identify these adducts, which are common. And we are building a user interface via web, so people can benefit from that and upload their data and start analyzing. So that's going to lead me to my project here at NIMBioS, which is to identify epitopes, MHC class 2 epitopes. It's different, and it's a challenge because I don't know much about immunology, and here Vitaly is going to help me understand the immunology. So I decided to do that because in my lab, remember I told you there is no vaccine for that? Well, in my lab they develop a vaccine that works on mice for this disease. One of my mentors, Igor, and one of or collaborators, Alexandre, they are working on that, and they developed this vaccine using a GPI molecule. And they use part of a protein epitope, but it's attached to a glycan. So it's an interesting molecule that you find that you have to produce the immunicity in the host. And for this project, they asked me to do a very short analysis, bioinformatics analysis, using tools to identify epitopes. And that's why I started to liking. And also, I like it because the variant surface glycoproteins, I think they are very fascinating because the parasite can switch and be undetected. Just a small reminder of the immunology. So an epitope is a fraction of a protein recognized by the immune system. These MHC molecules are glycoproteins expressed at the surface of nucleated cells. And T lymphocytes have T cell receptors that recognize epitopes only after they have been placed on the body's own cells by way of MHC molecules. These are the most common two classes of MHC, MHC class 1 and 2. MHC class 1 takes something that is in the cytosol, something that is wacky, something that is not working, and it sends a signal and presents some antigen and says, kill me because there is something wrong with me, like, for example, in cancer. And in MHC class 2 molecules, it presents some antigen derived from something that is exogenous, like a parasite. These molecules are highly polymorphic. In humans, there are more than 700 different alleles for this MHC class 2. And what makes the problem difficult? Well, this is a MHC molecule, this one here, and this is the epitope binding core. In the case of MHC class 1, the epitope binding core accepts up to 9 amino acids, from 8 to 9 amino acids. And it has a closed cleft, so it means that it can only accept those and no more. However, in the case of MHC class 2 molecules, it can accept, I have read in some papers, up to 23, up to 30, but most commonly is from 15 to 16. So it has open-ended, so the cleft is open. So there is a lot of variability in the peptide that can be here placed in this position. And also, one thing that complicates this is the primary anchor. The primary anchor is the MHC class 2 binding peptides contain a single primary anchor at the amino terminus, which, according to this author, is a hydrophobic amino acid. This I'm trying to understand better because this is something that is going to be important on determining what are the epitopes that I'm going to select to start classifying them. There has been some other work related to this. Positional scoring matrices, Gibbs samplers, HMMs, artificial neural networks, partial least square support vector machines, and some consensus. These algorithms work for only a small fraction of MHC alleles. The ones that work better are the machine learning algorithms, particularly ANN and SVM. And these algorithms have an accuracy of 80 to 90%, 70 to 90% accuracy. They are very good. So this is one of the challenges that Vitaly is asking me to see if I can do better. It's a good challenge. However, I'm not only proposing to classify the epitopes, I'm also proposing something else. This is the proposed method that I have. It's called logic minimization and rule extraction. I'm attacking this problem with two algorithms, but today I want to tell you about this one because it's my favorite. LMM is the application of algebraic axioms to a binary function in order to reduce the number of digital variables and or rules needed to express the function. The advantages is that they provide us sets of rules that can be used to predict peptide MHC banding. So the idea behind of LMM comes from electronics. In electronic, you have a circuit that it has a lot of connections, but then it can be minimized or simplified to something like this. And it will have less connections and it will be shorter. And this is what we see every day. We see that the devices are getting smaller and smaller and smaller, and many of those is because of this idea. So this idea has been around in electronics for a lot of time, but it has not been explored a lot in biology. And that's interesting that an idea like this has not been explored so much, at least in molecular biology. I have this example. These are some patterns. And imagine that these 4 variables here are amino acids. And then each one is a binary encoded, and it has an output. Imagine that this is a binder or non-binder. So this table and the other table are the same. Both tables have the same interpretation, and that is if b equal to 0 and d equal to 0 or c equal to 1, then the output is 1. Any other case, the output is 0. So you can minimize a great amount of information into just this short thing. A particular variable with symbol dash means that it's irrelevant to determine the output. That's why it has these dashes here. When we apply this, we have to encode the amino acids into 20 bits, 20-digit binary encoding. For example, letter A will be 1 and 19 zeros. The next letter is 0, 1, 18 zeros, and then the next 0, 0, 1, and so on. And predictive information outside the 9-mer core, which is almost up to 23 amino acids, will be represented by 460 binary variables after the encoding. And this is what represents a technical challenge, because existing software for doing this in electronics, it can only handle 22 input variables. So this is one of the things that I'm working now to see what can I do to do this challenge. So the goal at the end will be to build this set of rules, something like this. If amino acid 9 is C, and amino acid 12 is A, and 15 is G, and 18 is not I, then the peptide is an MHC2 epitope binder. And if you remember the omega site, which is important for the biologists to do either a drug or a vaccine, then this information is what they want. They want to know exactly which amino acid and in which position. They not only want to know if the epitope is binder or not, or how good it is. They also want to know something more relevant. And this is why this is my favorite algorithm, and I'm proposing to do it. So I want to develop rules that can classify epitopes and that are expressed in a language that can be understood by any researcher in studying this type of binding. As a testing data, I'm using this epitope database, IEDB, which contains antibody, anti-cell epitopes for humans and non-humans, rodents. My real data is from cruzi, which consists of around 64,000 proteins. Each one of these, I already have the GPI-anchored proteins, which are 4,825. I have predicted the proteins. And this is also a challenge, because imagine each one of these proteins, and each one of them contains around 300 amino acids, let's say. And if you want to separate only the ones that contain, let's say, from 9 to 16, all this range is millions of combinations. So all these millions of combinations require a lot of computation to do the classification and the rule extraction. So this also imposes a technical challenge. I am dividing these categories between binding and non-binding according to the IC50 value of the epitope. And the IC50 is the concentration of an inhibitor where the response or binding is reduced by half. So my model will be like this. The input the selected epitopes with binary encoding. As a training set, I downloaded the epitope alleles with these alleles H2IAD, which is mice, and this, which is from human. And then the output will be the categorized binding epitopes and rules for binding epitopes. Currently, I am doing the protein selection. I'm using only GPI anchor proteins, but maybe I will go to other BSGs. And because the software can handle only 22 variables, I might either I'm working on an algorithm to reduce the variables and get the most significant 22 and see if those can describe the data set, or to do this in parallel and work with all the 460 or so variables. Then from the predicted epitopes, according to the IC50, I might have to do a blast search or something to determine if these are self or foreign. You don't want an epitope that is already in you because it's not going to work. And then the 40 cruzi compare mice and human alleles. Mice and human because I'm working with a lab in Brazil that they have mice to do the experiments and to verify that the epitope in fact works. And they have serum from infected humans, and they can also test with the serum. And that's it. That's what I have. So I like to acknowledge my team in UTEP, my mentors, Igor and Mingying, collaborators, and my mentors here are Christina and Vitali, and they are my collaborators here. And I take any questions.